Okay, so now let's look at muscle contraction. We all know that one of the main functions of muscles is to contract in order to generate force and allow work to occur. And in our previous videos, we just saw how muscles are actually stimulated, and that's the way in which they get the signal to actually contract. So let's look at the actual contractile unit in muscles and see how that allows contraction to actually occur. So the contractile unit in skeletal and cardiac muscles is called a sarcomere, which we know has Z lines that form the boundaries of the sarcomere. And from the Z lines, we have the thin actin filaments radiating on either side. And we have the thick myosin filament interspersed in between these thin actin filaments in this conformation. Similarly, in smooth muscles, we have dense bodies which take the role of the Z line. So if we imagine this elliptical structure here being the dense body, it still has these actin filaments protruding from either side. So if we have this contractile unit in mind that's common to all three muscle types, we can understand how the general mechanism of the muscle contraction is common in all these three various muscle types. So before we look into the actual contraction mechanism, we need to look into a little more detail into the structure of actin and myosin. So let's start with actin. Actin is actually made up of several globular molecules called G-actin, which together form the filamentous actin called F-actin. So they form like a filament like this together. And each globular molecule has its own active site. And then for myosin, in a very simplified manner, just to understand how the structure affects contraction, we have the myosin head, the hinge, and the tail. And a bunch of these myosin molecules come together like this, and the tails bundle up together to overall form this thick filament we see over here. So we have the head. An important thing to remember about the head is that it has an innate ATPase activity. And from the name, we should know that it's an enzyme that hydrolyzes ATP into ADP and inorganic phosphate. And we'll see how that's important in contraction a little bit. Then we have the hinge, and that's important because it can change angles, which we'll see later affects contraction as well. And it's important to note that this head and hinge together are what's called a cross bridge. So keep that in mind. And then, like I said, there's a tail, which together bundle up to form this thick filament we see over here. So now that we have the basic structure in mind, let's look at the general contraction mechanism that's common to all muscle types. So let's say we have an ATP molecule bound to this myosin head. So let's redraw the actin and myosin, and let's focus on one myosin head just to illustrate how this contraction occurs. So let's say this one has the ATP bound to it. Now, since the head has an innate ATPase activity, this ATP would automatically break down into ADP and inorganic phosphate. So once this hydrolysis of ATP occurs, the angle of this hinge moves further to match up with the actin site further down from the previous actin site. So let's say that it had previously bound to this actin site, now it would change angles to bind to this actin site. So let's fill this up because this one's already done binding with. Just imagine. And let's say this myosin head now has ADP and inorganic phosphate bound. So when this angle change occurs, this conformation is actually kind of like a cocked position, is what it's called. And that's a high energy conformation because when ATP is hydrolyzed, it's a high energy conformation. And when it's in this position, the myosin head has a high affinity to the actin binding sites. So then let's say it attaches to the actual actin binding site. Let's say it's this one. And now it's bound to that, staying in that cocked position. So here we see that the ADP and inorganic phosphate are bound. The next step is this inorganic phosphate is released. And when this occurs, we get what's called a power stroke. What this basically means is that this angle of the hinge changes where it pulls the actin closer to the center of the contractile unit. So it would move, in this case, it would move the myosin head this way. And when it does that, it pulls this actin with it. So let's draw the fact that only ADP is bound when this power stroke occurs because the inorganic phosphate is released. Then the next step, the ADP is released. And this allows ATP to come in and take its place and bind to the myosin head. And this is important because this allows the myosin head to let go from the actin. So if we draw that, it looks like this. So it'll stay in that same angle, and just the only difference would be that it's not attached to the actin anymore. And so now we have the ATP bound in place of the ADP. And then what happens now is this ATP again hydrolyzes to ADP and inorganic phosphate, and the whole cycle begins again because that hydrolysis would again change the angle of the hinge. And now where the myosin head was attaching to this actin site, it's going to target the next actin site further down from it. And as you can see, as it pulls the actin closer to the middle of the contractile unit through these cocked position and power stroke and release of ATP, all of this together allows the filaments to slide past each other. And that's why this is called the sliding filament mechanism. Or sometimes it's also called the walk-along theory because it looks like the myosin head is walking along step by step from each globular actin molecule active site to the next. So the whole point of this contraction is to bring the Z lines or the dense bodies to come closer together. And you can imagine how that allows the muscle overall to contract and shorten its length. And it's important to remember what zones in this contractile unit change because of the sliding filament. So if you recall from our structure video, this would be the H zone. And then this would be the I band, since it only involves the thin filaments. And this together, the thin and thin filaments together, would be the A band. So if you imagine the Z lines or the dense bodies coming closer together, this H zone would get smaller as it comes closer. And the I band would get smaller because more of this green actin would overlap with the blue thick myosin filament. So during contraction, the H zone and the I bands get smaller. They can also actually disappear as the sliding filament mechanism continues.